Thanks everyone for attending. My name is Maxim Jacobs. I'm Director of Healthcare Research at Edison Group. So just thought, let me, thank you to all the panelists for being here. So let me just start off by asking a question. It might be a little bit of a big question, but it's for the KOLs. I was just wondering, can you just briefly summarize the pluses and minuses of the major classes that currently treat hypertension?
Well, there are 125 different blood pressure medicines. The major classes are diuretics, calcium blocker, renin-angiotensin blockers, which are ACE inhibitors, and angiotensin receptor blockers, the beta blockers. And then there are some other medicines that work centrally, but they're medicines which have a lot of side effects. So let me choose 2 or 3, and then Henry, you can take the other classes. Diuretics are the first step in most patients. They remove excess sodium. They also help remove an excess volume. They have the unintended consequence when you have the use of high doses of diuretics, it lowers potassium, hence the cramping, the weakness, low magnesium. It also can elevate the uric acid, which can cause gout in some patients, not as commonly. Does not have a beneficial effect on lipids, but there are good blood pressure medicines, especially in middle-aged and older patients, African Americans, patients with diabetes and chronic kidney disease. So that's why we added, as a second step, if needed, a low-dose diuretic. The calcium channel blockers block the constriction of the blood vessels by blocking the influx of calcium. Calcium causes smooth muscle that wraps around the blood vessels to constrict. They also have a mild diuretic effect and appear to be also very efficacious. So hearing that leaves the RAS blocking agents, the beta blockers, and the central blockers.
Okay. Thank you, Keith. It's hard to categorize what we currently have. I tried to do that to some degree. And diuretics as well. There are subclasses of diuretics. Aldosterone blockers, for example, had a diuretic effect, but they're also anti-aldosterone effects. Beta blockers and alpha blockers work in the central nervous, in the nervous system, usually in the peripheral nervous system, and they also have effects such as lowering heart rate if you're a beta blocker, but they can cause fatigue. And a lot of people on beta blockers are not very happy with them, and they feel much better when you stop their beta blocker. Then renin-angiotensin blockade can come with ACE inhibitors, or they can come with angiotensin receptor blockers. ACE inhibitors were originally discovered from snake venom about 50 years ago, and they interfere with the renin-angiotensin system in the conversion of angiotensin 1 to angiotensin 2. The angiotensin receptor blockers block the conversion from angiotensin to the attachment to the site of action, which is in the blood vessels, and they tend to be better tolerated. ACE inhibitors can cause cough in a substantial number of people, and that's a problem. And I think a lot of people who are on ACE inhibitors feel better when they're off them and on angiotensin receptor blockers. There are dozens of examples of those 2 classes of drugs, which for the most part are very popular and more popular than diuretics in a lot of people's hands. If you deal with an older population, ACE inhibitors and ARBs, as they're called, are not as effective as diuretics and calcium antagonists are. And calcium antagonists come in 2 buckets also. Dihydropyridines, which can cause swelling, ankle swelling particularly, and non-dihydropyridines, which slow heart rate and are often better tolerated. There are only 2 of those. They were, in fact, discovered back in the 60s, and there have been no new ones since then. Diltiazem is the second, verapamil was the first, and as far as I know, there's never been any further. Then there were drugs that work on the central nervous system. Alpha-methyl-dopa was the first of those, and then there's a drug called guanethidine, which I think I used a little bit in 1967, but almost not since. But for the really resistant patients, you've got to concoct a cocktail, which includes several drugs. At one time, it was felt inappropriate to use more than one drug at a time. You'd use one, if it didn't work, you stopped it and used another one. We've long given up that paradigm, and now we'll add drugs. And sometimes we start with 2 drugs. As I showed you in JNC 7, the recommendation for people with what we called stage 2 hypertension to start with 2 drugs, knowing that the likelihood of getting their blood pressure to goal with one was pretty slim. So that's pretty much the group. We have lots of choices, and in spite of that, we still leave about half of our hypertensive patients not at goal. And I don't like the word controlled, I'd rather say at goal. And those goals are a moving target also. For diabetics, we want it to be less than 130 systolic, and for non-diabetics, less than 140 systolic. Now, you see some people, some practitioners, when you have somebody on a regimen that they tolerate and their blood pressure is 112, back off. That's exactly wrong. As long as you're mentating and urinating, your blood pressure is high enough. And I think that's how we ought to look at things. So if you get down to 112 over 70, congratulations. That doesn't mean you should have less drug. It means the regimen you have has been effective, and you should stay with it.
Great, thank you. That was very helpful. Now, you mentioned how you tend to use combination therapy for patients. So when you do add like another drug into a cocktail, like what do you consider a meaningful benefit from that drug?
Well, first of all, because it may be confusing, because we're studying a first step agent and then adding the diuretic. And the reason we're doing that is because you want to demonstrate the effect of the medication in a low renin, high-risk patient population, and you're adding the diuretic if needed. So you don't want to start with combination in a research trial with a new agent. That being said, in usual clinical practice, you balance out some of the side effects of one medicine by adding at a relatively low dose to another medicine. So for instance, with the diuretics, the complications of wasting the potassium can be blunted by adding it to the renin-angiotensin system blockers, such as an ACE inhibitor or an aldosterone antagonist or an ARB. That's why a lot of your combinations will have, for instance, valsartan hydrochlorothiazide or lisinopril HCT hydrochlorothiazide added together. But that is for therapy. That's not research. In research, especially with a new agent, you like to see the effect of that agent before you add the other medicine.
Mm-hmm. And what sort of effect would you like to see with that, with like a first step agent?
Any first step agent?
Or like a new one that's coming, the new kid on the block?
Well, one of the questions, which was an excellent question from the audience, was why the p-value was not statistically significant. And I think Bruno answered that appropriately, is that you're talking about 30 patients versus the study that, well, I actually designed much of the study. I'm not bragging, but I did. We're talking about 250 patients. So it's exponentially a larger sample size. So that same effect, which didn't appear to be large, you would agree, Bruno, in a larger sample size may have been very statistically positive.
Yeah. Yeah, no, but I was more looking for just like clinical significance. What sort of level are you looking to see?
When you look at populations, small degrees of blood pressure lowering have profound effect. So for instance, in the all-hat trial that I showed you, where I said that chlorothalidone, a diuretic, was better than lisinopril in the black cohort, the difference in systolic blood pressure was 4 millimeters of mercury, black versus white. Doesn't sound like a lot, but in a large group of people, 3 to 4 millimeters of mercury has been shown to have profound effects on heart attack, strokes, and overall cardiovascular disease. In an individual person with a first step agent, hopefully you would have a little bit more than that in order to show a statistically significant effect. But in a population-based study, 4 to 5 millimeters of mercury is a robust difference.
Okay, great. Thank you. So just want to maybe open it up for questions. If you have a question, please just raise your hand and a microphone will come to you magically. No? Okay.
Answer this question.
Okay. Wait,
Yeah,
just-
wait for the microphone.
Dr. Ferdinand, Jason McCarthy at Maxim Group. I know you kind of answered that question on millimeters of mercury difference, but overall, would you expect to see something greater than 5 millimeters of mercury difference for a new drug as a monotherapy, particularly if you go up in baseline blood pressure to 160, 70, 80? You know, we saw the curve that you showed on slide 12, I think it was, where it was widening. Your thoughts on that, and you know, how do you address that with the patients that you're enrolling in this upcoming New Hope study?
Yeah, that's an excellent question. The higher the blood pressure, the bigger the fall. And you would expect in a population which has more racial ethnic minorities, more obesity, to have a more salt-sensitive population. So we're kind of sub-selecting for patients that may have not shown an effect in the 2A phase study. Higher blood pressures, more racial ethnic minorities, more obesity, which is a surrogate for salt sensitivity. Salt sensitivity means that if you infuse sodium chloride or you give the person a high salt diet, their blood pressure goes up. We're not measuring that because the observational data indicate that this population tends to be salt sensitive. So we're kind of using the phenotype as a surrogate for measuring salt sensitivity. Now, that being said, what one would hope, I hate to predict studies before they're done, but you would hope a more robust individual blood pressure lowering, especially with the higher blood pressures in this selected population, and I would hope that it be well above the 4 to 5 millimeters of mercury. Even if statistically significant, a first step agent, which is very effective, should be as much as 8 millimeters of mercury or even a lot higher.
You have to be careful for a couple of concepts. One is called regression to the mean. So if you really start very high, the likelihood you'll be falling, that has nothing to do with the treatment, just a statistical quirk that you have if you're high to begin with. So that's something that a larger sample size should take care of. You have to calculate a sample size based on your predictions of how much the blood pressure will drop and how different it will be between two groups, and that's where the statisticians come in. I'm a rookie with statistics, not even a rookie.
We have had a
incompetent.
statistician look at the sample size, and it predicted with the 250 millimeters of mercury that we would get a significant lowering as much as 6 to 7 millimeters at least.
The most important thing when you design a trial is to calculate the sample size properly. And to do that, you need the estimates of what you've got, which I think we have here to start with, but as the trial gets larger, we have something called stopping rules in every trial that we do as well, where if you see certain amounts of events, you stop the study because it's potentially harmful to one of the groups. And sometimes you stop studies for futility, where you're so far into it, you're never going to be able to prove what you did. And all those things make clinical trials a pretty interesting discipline right now.
In the back, there's a question.
So in the upcoming phase of the trial, will there be any of those second step, like a diuretic, during the trial?
Yes, the diuretic is used for patients who don't have a blood pressure at, is it 4 weeks or 6 weeks?
So they will
4
remain
weeks.
on that diuretic during the trial?
During the trial, yes, it's added for patients who have not had a response to increasing dosage from the 250 BID to 500 BID at 4 weeks. And the diuretic we chose was hydrochlorothiazide. The dose we chose was 25 milligrams. Those of you who know medicine know that that's not a very powerful agent. We purposely did that because that's what most clinicians still use as their diuretic of choice. And many of the branded medications and commonly used medicines use 25 milligrams of hydrochlorothiazide. So we wanted to do something which would somewhat reproduce what is used in usual clinical practice.
Now, in all hat, we use chlorthalidone, which those of us who
Better.
know the field feel it's a clearly superior drug. Hydrochlorothiazide and chlorothiazide came out first by a big company, a powerful company, also known as Merck, and they marketed it very successfully. Chlorthalidone was made by USV, which was a small company, but the NIH liked it better, And the NIH trials will use chlorthalidone, which I think most of us have realized is a much superior thiazide-type diuretic.
Right, but Lionel and Brew and I discussed this extensively, Henry, even in some discussions that you weren't part of because they weren't part of the formal structure. And we just thought because of the wide use of the hydrochlorothiazide, the readily available use of the hydrochlorothiazide, and the relatively safety of hydrochlorothiazide that we would use that.
Yeah.
I agree with you, chlorthalidone is a more potent, more effective thiazide-type diuretic.
Great. The gentleman in front here who asked a question.
Dr. Ferdinand.
Wait, you can just wait a second for the microphone.
If someone has a history of atrial fibrillation, does that throw off your study?
Yes, and I'm going to turn to Bruno, who's the chief medical officer. What are we going to do with patients with atrial fibrillation?
We won't exclude, in this trial, we won't exclude patients with AF, atrial fibrillation, yeah. We have done in our heart failure trial, but for other reason, because atrial fibrillation has an effect on pro-BNP, which was the primary endpoint, but this is not the case here. So patient with atrial fibrillation, and especially atrial fibrillation is very common in elderly, and we will select hopefully elderly patient as well. So we will have patient with AF.
Just in terms of, the type of patient, what type of patient would you consider giving, QGC 001 to, assuming the data, is positive?
Well, Bruno showed the pre-human data that suggested that a salt-sensitive type of patient, a DOCA salt-sensitive type of patient, would best benefit. And as I mentioned to you before, there have been studies that look primarily at African Americans, patients with diabetes, patients who are older, patients with obesity and overweight status who tend to be more responsive to that type of medication. So what is that type? Those are diuretics and calcium channel blockers. They're not ACE inhibitors or angiotensin receptor blockers, so it's not lisinopril, captopril, valsartan, losartan. In fact, there have been some studies with outcomes in the patients on the ACE inhibitors and on losartan, the black cohort actually did worse. Now, that doesn't mean you don't use those medications. It just means that you often have to combine them with the diuretic and the calcium channel blocker. But because this is a proof of concept study, we're not going to use combination to start. We're going to self-select those patients based on the variables that Bruno described so eloquently, and then see how those patients respond with the backup at 4 weeks of the hydrochlorothiazide. In usual clinical practice, what I would expect, that if we can show safety and efficacy as monotherapy, patients are going to still need a combination. But you wouldn't want to start with the combination because then you take off the table those patients who respond to monotherapy, and you take off the table those patients who may respond to combination with another agent, chlorthalidone or amlodipine. So we're not going to start with the combination. But in usual clinical practice, I think Henry kind of said it, but I'll even say it clearly, most patients end up with combinations.
It's a curious thing, too. You get assigned in a trial to what drug you start with, even if you only take one pill. That's the group you're assigned to. Now, that doesn't really make sense, but that's how we do it, and sometimes that can confuse the outcome. And so you have to start cutting through and digging deeper and deeper into the details of the trial, which hopefully we'll have in the 250 pretty soon. But if you get assigned to the agent and don't take more than a pill, and because of a problem, even though something else is used, that's going to still be what was blamed or credited for what happens.
Let me answer a question that no one's asked. Why we don't have a placebo arm? None of you thought about that? When you have a new drug, in the past, if you go into the 1980s, the 1990s, they usually do a placebo arm. We talked to FDA already. We've met them in person. And it is so well now accepted that treating blood pressure is beneficial. They don't need a placebo arm, especially if you're doing the 24-hour ambulatory blood pressure, which is an objective way of measuring the blood pressure. They would suggest, therefore, that if you show benefit, there is a benefit. And you could push for a placebo-controlled trial, but they did not request it. That's a kind of difference if you look at some older studies. If you just research blood pressure studies and you went back into the 80s and the 90s, you'd see a lot of placebo-controlled trials. I wouldn't want to be in a placebo arm myself.
And in the previous study, in the phase 2A study, we had a placebo group, and in the placebo group, there were no change in blood pressure during 4 weeks. So it validates the fact that we won't have a placebo group in the next study.
Yeah, but we did ask the FDA in person
Yeah, we did.
about
We
that.
did. We even proposed the FDA with a design with a placebo group, and they said, "Why do you want to include a placebo group? This is not useful."
It's a curious thing, too. The British Society recommends or that anybody that you diagnose as hypertension should have an ambulatory blood pressure monitor. It's interesting to find that the British want to spend more money than we do, but that's what you see. And I don't know that we could ever really implement something like that here. There's still a lot of very good centers that don't have ambulatory blood pressure monitoring
Absolutely.
available.
I think the fact that we're doing the ambulatory blood pressure monitor, although the clinic blood pressure still is the primary endpoint, an important secondary endpoint will be the ambulatory blood pressure monitor.
Now, you had brought up the FDA, a second ago, and I was just wondering, the FDA seems to have become more relaxed, in the last couple of years, and I think the new FDA commissioner also seems to want to become more liberal. Now, in the past, hypertension medications were approved with cardiovascular outcome studies as well. Do you think this could be approved in a phase 3 just focused on hypertension without a pre-approval cardiovascular outcome study?
Yes. So to break down your question for those who are non-scientists, often the FDA in the past will say you have to prove that you decrease heart attack, strokes, or kidney disease before we approve the drug, that you have some benefit outside of just blood pressure. But the data have been so convincing, so consistent, that if you lower blood pressure safely, you decrease heart attack, strokes, and kidney disease, that the FDA no longer requires that. So there is not a need to do an outcome study before you get approval for a new blood pressure agent.
Great.
And they have approved an agent, herbicide was approved within the last 5 years, and it had no outcomes.
That's sort of different from what happened with diabetes drugs, where the FDA mandated an outcome study to be done while the drug was available, and we're beginning to see those results now.
Well, that's because the paradox is lowering glucose has not been shown to be as effective for decreasing heart attack, strokes as lowering blood pressure. Blood pressure as a surrogate actually is a more powerful predictor of risk than glucose. So those of you who have diabetes or you have a loved one with diabetes, it's important to control glucose, but perhaps the best thing you can do to stop them from having a heart attack, stroke is to control blood pressure.
I mean, I think also just in the diabetes medications, a lot of them had such side effects that whatever benefit they were getting from the glucose, they were losing from the drug.
Right.
Yeah. Yeah, so maybe I can ask a basic question on kind of treating hypertension. I think you, Dr. Fernand, you mentioned that you tend to want to treat systolic versus diastolic. I was wondering if you could just explain that a little bit.
Yeah, I'll do it as simply as I can. As we get older, the vessels get stiff. So when the heart pumps blood, it runs quickly through the vessels and then bounces back. So the systolic, which is the measurement of that pressure that is found when the heart pushes the blood through and bounces back, is more predictive of heart attack, strokes, and dying than the lower number, which is a measurement of the runoff number. And in fact, in older persons starting about age 55 and above, if you looked at systolic blood pressure, you looked at diastolic blood pressure, systolic blood pressure is more common, even with a normal or low diastolic, and systolic blood pressure is a better predictor of heart attack, strokes, and dying. So the FDA would probably not be as accepting if we showed diastolic blood pressure reduction, but we didn't reduce systolic blood pressure. They want to see systolic blood pressure. So as a primary endpoint, clinic systolic blood pressure, and then a follow-up 24-hour ambulatory blood pressure looking at mean systolic blood pressure and nocturnal systolic blood pressure, I think would be favorably accepted by the FDA versus looking at diastolic blood pressure.
The more basic question is, why was it diastolic in the first place?
Why
Both
was
of
it
them were.
diastolic in the first
You know, why pick diastolic or systolic? They were both available. I think the idea first was that diastolic was, the blood vessels were in diastolic pressure longer. And I think it was a question of, and I've had this confirmed by some of the people who were there, you see somebody walking into the emergency room with a blood pressure of 160 over 120, having a hypertensive crisis. And then the next patient you see is a so-called LOL, or a little old lady, who's 230 over 60 and doing fine. So the diastolic is probably what's important. And I think that sort of carried through until people began to realize that both of them were important. If anything, in the older age group, systolic is a much better predictor of everything that you see.
Yeah, I'm very confident that we chose the right endpoint
Yeah.
using systolic blood pressure.
In fact, I'm not even sure we should measure diastolic blood pressure.
Not going that far. I'm very confident we used the right endpoint.
We'll still do it because we're very tradition bound, but you learn very little new from if you don't know the systolic also.
Now, just in terms of actually measuring the blood pressure, what percentage of the time is the blood pressure read incorrectly because of like poor placement of the?
Most of the time, it's done inappropriately.
Yeah, often.
And then what sort of error does that result in?
Significant.
You get an inaccurate reading, and you based everything on the numbers you see, and they're not accurately measured. Then you're kidding yourself.
We presented a lot of data here today and a lot of studies. And Bruno, if you want to illuminate, you may, on using the sprint method that we're going to use.
Yeah. So first of all, it's always easier in a clinical trial to measure the blood pressure appropriately as compared to clinical situation, because we provide the investigator with a procedure for that. We have chosen the office blood pressure and the sprint-like measurement. So it's in between the common office blood pressure made by the physician and the self-measurement. What will happen is that the physician will put in place the cuff for measuring the blood pressure and leave the patient alone, and the system will take the blood pressure every minute, and there will be an average of 5 measurements. So it should avoid the what's so-called the white coat effect. The patient is afraid of the physician who takes the blood pressure, and by averaging, we will decrease the variability. But we will also have the ambulatory 24 hours ambulatory blood pressure. So we expect that at the end, all the results will be consistent, whatever the office blood pressure, sprint-like, or the ambulatory blood pressure.
One other kind of probably more basic question I have is, in the chart of where you showed the baseline in the trial, it was obviously lower at night. I mean, is the nighttime blood pressure something you should ever even take into account? Is it important?
Yeah, it's very important. The persons whose blood pressures don't lower at night are exposed in the vascular system to pulsatile pressure for a longer period of time, and observational data says that they're at increased risk.
We call those people non-dippers. That means their blood pressure doesn't fall, or dippers, which is what most of us are, and they have a better outcome.
How many are what percentage are non-dippers?
Depends on the group you're looking at. In African Americans, it's pretty high. I wouldn't.
30–40% at least.
And in people with, say, heart transplants, it's very high. There's no change in blood pressure at night. But I think for most people, younger people especially, it's about 10 to 15 millimeters of mercury at least, the difference between day and night. That's the sort of information we get from an ambulatory blood pressure monitor, where you see how much it falls at night.
And this is what happened in the phase 2 trial, because we did not select the population, and if you remember the graph, the blood pressure decreased at night, and also this is probably why the effect of QGC 001 at night was little as compared to what observed during daytime, because the blood pressure was lower or even normal, so we did not show any effect of the drug, which is expected based on the mode of action.
You can't lower if it's already where you want it.
Of course.
So in this study, we would expect to have more or an increased prevalence of non-dipping, which has the benefit, if a drug works at night, to show more benefit.
Okay, great. Brad, over here in the front. Yeah.
Thank you. This question's for Dr. Ferdinand. Can you speak in a little bit more detail regarding the change in inclusion criteria for the phase 2A to the phase 2B, specifically as it relates to what seems to be a rather modest increase of baseline blood pressure from 140 to just 145 in the context of your best responders in the phase 2A being those with baseline blood pressures of 170 or greater?
Thank you for the question. There's a safety issue that Bruno may also want to address. You will probably have a better benefit with higher blood pressures, but at the same time, you don't want to have them so high that the investigators are resistant to enroll patients. Now, if we used 140 as the cutoff, then you would have patients who may indeed have labile hypertension, and when they got into the study, they may not have much benefit because their blood pressure was kind of bouncing around as it was. So you want people who have fixed blood pressures that are elevated. We used 140 as a somewhat arbitrary cut point to have safety, but also have potential efficacy. Whereas if we had just said, well, let's push it all the way up to 160, you may have shown more efficacy, but the investigators would have a sense of less safety. The FDA also may have frowned on entering such a high level of patients' blood pressures for a first study in the United States. So it's a lot of mixed factors that go into that. That's where we came up with the 145 to 170.
So, my next question is just really about, what will happen, when QGC 001 is on the market. Do you usually receive pushback from payers to use branded medications for hypertension, given that it's generally a generic market?
You went very far into the future. The general rule of thumb is in managed care, which almost all of us are in managed care now, unless you have what's called an indemnity policy where you go anywhere you want, pay whomever you want. In managed care, where the company says you can go to this group of doctors and you can get this group of medications, and if you don't go to this doctor, you pay a penalty, and if you don't use this group of medicines, it's going to cost you a lot. They usually like you to use generics first. I could see, and again, we're way in the future, we're years down the road, I could see in a patient where you could demonstrate they did not respond to an ACE inhibitor or an angiotensin receptor blocker, and you wanted to use that class of medicines, that you could postulate and you could write to the managed care corporation, I'd like to use this drug, which has been shown to be efficacious as a surrogate.
Would you expect a lot of pushback? I mean, I know a lot of them will want the step therapy, but if you just document it, do you think that will be enough?
I have clinic twice a week, and I can tell you with some of the new agents, like the new injectables for cholesterol, et cetera, they push hard against you being able to use them. And you not only have to document and fill out a paper and fax it, and you often have to do what's called peer-to-peer review, where you have to call a retired eye doctor in Iowa and talk to him why you want to use a PCSK9 inhibitor. It gets very, very tortuous. And that is purposeful, because they want to block you from using the newer agents. I think the struggle may be that if you have a newer agent, it has to be cost effective, and you have to negotiate with the managed care corporation through the pharmacy benefit managers. They're people who decide which drug, such that you may have to do a loss leader and sell your drug at a cheaper price than would be on the open market. It's a lot of complications with new drugs.
Yeah, but I mean, I think PCSK9s are, I mean, they are extremely expensive, especially compared to what, you know, Quantum Genomics will likely price it at. And I remember seeing that it was something like $2 million per prevented heart attack.
They've done cost analysis, and they seem to be beneficial.
Okay. More questions? Now, you know, how many patients with, like, you know, low renin hypertension do you guys currently treat in your practice, or is it something that's not generally measured?
We usually don't measure renin. We're here in New York City, where at Cornell, John Lara, the late great John Lara, measured renin in patients and found that those patients who had high renin responded better to ACE inhibitors and angiotensin receptor blockers, which are typically not the minorities, not the older persons, not middle-aged women. I'm at Tulane University in New Orleans. The city is predominantly African-American, have some Hispanics. The patients that I see, most of them I would probably consider low renin. I just don't take the time to measure it, because the way you find out is you put them on certain medicines and stand back and see how they respond.
Well, I happen to come from the other camp. I think that was just
He's from
one.
New York City.
No, this is New Haven, where I was at the time. Barry Madison did a VA study where he did measure renin the way John Lara wanted us to, and it turned out knowing the renin level was no better than knowing the race and age of the patient. So it was a nice theory, but it doesn't really help you, and I'm very happy that you're not doing it. I would argue if you did.
No, I don't do it.
Good, because it's just not it was a good concept. I think it did help to categorize hypertensives in various ways, but that's not the best way. Age is a better way, race is a better way, and a sophisticated clinician will be able to select what looks like the best drug.
So that's why it's, you know, renin level is not a requirement in the upcoming trial.
Absolutely not.
Yeah.
Great. And then, sure.
When you're powering the phase 2 study for 250 patients and you're looking for a delta or drop in millimeters of 4 to 5 would get you a p-value, is that based on what you saw in the phase 2A study, assumptions around the reduction in mercury of 2.7 to 4.5, or does that take into account kind of sweetening the pool of patients with low renin or high salt, like an African American population or a diabetic population, where that delta might be much wider? Can you just...
It was a second. We were aware that the phase 2 study done in Europe did not have the type of robust numbers, but we felt that if we had a more predictive population, we would get better numbers, and then the statisticians backed us into the 250. Go ahead, Phil.
You saw the All Hat study, which I was a major participant in. We had 42,000 people in that study. We wanted 50% African Americans. We had 35%. The reason we had 35% is we couldn't get 50%, not because they weren't there. And so if you have good statisticians, you plan a study based on what you anticipate. If you guess right, you'll have the right sample size. If you guess wrong, it might be too small, and then you won't be able to tell what you wanted, or it could be too big, and then you're wasting money. So it's a very important part of every clinical trial is how good your statisticians are and what data they have to work on. You've got some information from the small study, which will help you plan the bigger one, but I'm sure this is going to be looked at along the way and see if that has to be adjusted. You may need more than 250, or you may get your answer with 175.
Yeah, we just don't know. This is true research. One of the things you know, since you were taking excellent notes, is the European study was predominantly male, 57 years of age, lower body weight than what you're going to have in the United States. In the minority community, black men don't go to the doctor. They don't go in clinical trials. So paradoxically, although women are underrepresented in most studies, in the African American community, you have more women than men. So I suspect we're going to have middle-aged, older, overweight and obese black women who will respond. And that just goes from doing 100 different trials and knowing who comes in and who agrees to these studies. You can go to the barbershop and the ballrooms and try to get the men, but it's real difficult to enroll men. Whereas in the European trial, I think you were 73-point-something% male.
Yep, right.
Right, so your powering assumptions were based on like the all-half study, where like maybe there's 30% African Americans. Is that was my question. Is like, what assumptions are you making about the expected percent of different groups?
We wanted over 50%, more overweight, obesity, more African American, other minorities, maybe 20 to 30% diabetes. So versus 2 to 3 millimeters, we're expecting 4 to 5 at least. Those are assumptions, and assumptions of trials are often wrong. I hope that we're not wrong.
In the Women's Health Initiative, which I was also part of, we started from 50 and went to 79. As soon as we felt we had enough younger women, we stopped recruiting people. So 50 to 54 was eliminated right away. They were easy, so-called low-hanging fruit. And then we had to try to get older women, which was much more difficult to do.
And we took into account as well that the treatment period will be 8 weeks and not 4 weeks. And what we also took into account is the variability, the standard deviation coming from the previous trial. Because as you know, the power calculation is not only the minimum difference you want to show, as well as the power, but you absolutely need the variability, which is the standard deviation.
I'll ask one question, which no one did. If a particular subject, and I don't think they should be called patients, they should be called subjects, does very well and wants to get the medicine afterwards, are you prepared to do that? That was the question.
Yeah.
It's an unexpected question.
No, it's a difficult. Yeah, we could do that.
Some IRBs may want you to address that issue.
If we are starting the development, so sometimes we are a little bit short of treatment. So of course, for very few patients, we could be able to do so, but not as a general rules. At least for the time being.
I think all the IRB would want you to address that with a sentence. Unfortunately, we will not be able to provide medicine after the study's over. And that's all they want is an answer.
Yeah.
Okay, I thought maybe we can pivot to heart failure a little bit, and just I was wondering if our experts could discuss, how do the pharmacological needs of heart failure patients differ from, like hypertensive patients?
Heart failure is a very difficult situation. We are, as well as in hypertension, there are a lot of drugs, and especially new drugs, working quite well in terms of reducing cardiovascular event, and we are talking about mortality. And so this is another story. But still, the prognosis of patients with heart failure is not so good, even when treated with standard protocol, which includes beta blockers, ACE inhibitors, and as well as Entresto now. And interestingly, the very good result of Entresto in terms of reducing morbid mortality showed that the existing treatment were not enough. So there is still a need. The very interesting question for the future, but not especially for QGC 001, is at a certain time we will have to ask the question, should we add another drug or replace an old one? So this is another question. But to come back to QGC 001, first of all, aminopeptidase A is overexpressed in this patient, so blocking the enzyme would be beneficial in this patient. And we have shown a very interesting effect in a rats model again. And the effect was similar or a little bit higher on the cardiac function, the ejection fraction, as compared with enalapril, which is the standard ACE inhibitors. And by combining the two drugs, there was an additive effect in animals. So that's why we want to start the development in heart failure. And in the current trial running in Europe, QGC-001 is used on top of the existing medications, including Entresto when available, which is not the case in every European country.
I just want to add a comment on that. We did this study, this very exciting study on heart failure on animals with Professor Franz Linen with University of Ottawa. And when you look at the mechanism of action, I mentioned that not necessarily the same part of the mechanism of action that is more important in heart failure. And for heart failure, what Franz Linen has discussed at the very beginning of our program is that after a myocardial infarction, you have an hyperactivity of the renin-angiotensin system at brain level, and also an hyperactivity of the sympathetic nerve activity, an overexpression of the sympathetic nerve activity. So as our mechanism of action is targeting the renin-angiotensin system at brain level, but also decreasing the sympathetic nerve activity, when he contacted us at the very beginning, he said, I think it may be beneficial for patients that have chronic heart failure, and I would like to test your compound in animals. And that's where, at that time that we started this program on heart failure. And you're right, we have demonstrated in animals that we have a better improvement with our product than with existing drugs, such as losartan as well, and a synergistic effect with an ACE inhibitor. So that's very, very exciting, the heart failure program. And we can imagine that we are very happy to have this phase 2A study running in Europe for heart failure, and we can stand to wait for the results by next year.
Just can you remind everyone what are the endpoints for that trial?
It's a very technical endpoint. It's a decreases pro-BNP, which is something we measure in blood and is elevated in patients with heart failure. So it's a surrogate endpoint that is used mainly in clinical trial. It starts to be used in clinical practice as well. But once again, because this is a 4-week treatment, we won't be able to collect. We will collect, but to have an effect on clinical events, and it's also too short to have an effect on ejection fraction or feeling pressure. So that's why we have chosen this surrogate endpoint. And if we are successful, then we can be able to design a further trial with more clinical endpoint, or at least ejection fraction.
Mm-hmm. How long would that trial need to be for ejection fraction?
At least 3 months for ejection fraction.
Just add, because heart failure is very important as well.
Yeah.
Just want to add something. As you have seen on normotensive patients, we do not have a decrease of blood pressure. That's important for heart failure as well, because a lot of patients have hypotension. So having a product that is not decreasing blood pressure for those patients may be very beneficial as well.
And another point, but we are talking about more severe heart failure patient who is hyponatremia, so sodium level very low. And the reason why there is hyponatremia in this patient is because of high level of vasopressin, which is called sometimes antidiuretic hormone. And QGC-001 is decrease the vasopressin, so may be beneficial in very severe patients as well. But it's an hypothesis.
Dr. Ferdinand, in conjunction with the use of diuretics, you lose a lot of electrolytes at that time. Is anything done to actually add to the electrolytes that are lost?
Yes, in our study, they have looked at all of the preclinical data and the human data to date, and you don't get the potassium lowering that you see with diuretics, and you don't get the increase in potassium that you see in some patients who have kidney disease with an ACE inhibitor. So we would expect that because hydrochlorothiazide is a fairly mild diuretic, to not have any significant potassium shifts. Shifts meaning change. But as a precaution, we will allow the investigators to measure serum potassium if they're placed on hydrochlorothiazide after 2 weeks, to document whether the potassium level has dropped unexpectedly or maybe even increased. But as monotherapy with the quantum genomics medication thus far, you've not seen significant changes in potassium. In usual clinical practice, what we do is start the person on the diuretic, tell them to restrict salt intake, and then probably anywhere from 7 to 10 days after you start, tell them to go to the laboratory and get the chemistries checked. And if you see those shifts, we sometimes will add an ACE inhibitor, an angiotensin blocker, or spironolactone, or if it's just a mild shift in potassium, we may just give them a prescription for potassium chloride. It comes in 10 and 20 milliequivalents. The eat a banana thing is probably overblown. There's only 1 milliequivalent potassium in every inch of a banana, and you get the extra calories. So I would rather just have them take a low-dose potassium supplement than tell them eat bananas.
Great. So does anybody have any more questions? No. Well, thank you so much for all of your time. Lionel, I believe you'll have some concluding remarks.
That's a very funny story about potassium, which bananas anyway.
So thank you very much for. Yeah. Thank you, Keith. Thank you, Henri, for your talk. Very appreciated talk today. Thank you, Bruno, of course, and so thank you all of you for the discussions that were very, very interesting. I have just several remarks I've written during the discussion, and I just want to emphasize on those one. Henri Black told us that high blood pressure is due to several causes, not only being black or white, but also lifestyle. Also, there's plenty of reason to suffer from high blood pressure. And despite all the drugs on the market, we still need new class of drugs, and new agents will help the physician to do better in the treatment of the patient. And Keith Ferdinand too told us that not only the deaths, because you have seen the increased risk of death when you increase the level of blood pressure, not only the deaths, and we used to talk about how many patients are dying every year due to the complication of high blood pressure, but also the disability that was caused by the disease is very important to take into account. And when it comes to stroke, which is one of the most important complications of high blood pressure, I'm quite sure you all are aware about what disability means in that kind of case. New Hope, because hopefully that's the name we've chosen for this trial, because hopefully, and I'm reading what I've written because it's Keith's word, we have something new to propose to patients and physicians, and I think that's very important. QGC-01 has proven to be well tolerated in patients. Of course, in healthy volunteers, we did single ascending dose, multiple ascending dose in healthy volunteers. It's a common clinical development for such a drug. So well tolerated. We've shown an efficacy in animals but also in humans, and we kind of mimetic between the two different with animals and humans. And we have this increase of efficacy when there is an increase of blood pressure. And we plan to demonstrate with this New Hope study that due to this unique mechanism of action, with this kind of triple mechanism of action, targeting the brain to act on vessels, heart and kidney, we may be beneficial for patients that are today suffering from those conditions, that are dying for those conditions, but also having complications turning into disability. So I think that's the key words for this this KOL day today. And I remember at ESH last week a physician giving a talk about the percentage of patients that are well controlled. Sorry, Henri, about that, but it's not my word. But that's not at goal, we say, because you're right. The goal is not the same when you are suffering from diabetes and high blood pressure, and when you have other additional risk. But you say, well, there are only 50% of the patients that are well controlled by existing drugs. So what do we do now? And I would say, well, we can invest more and more money in innovation in that area of business, because there is a huge unmet medical need. Thank you very much.